My name is Marianne Stanford. I'm born and raised in a small town in Newfoundland. I've been a postdoctoral fellow for about 5 years, and a postdoctoral fellow is someone who's completed their PhD but needs to gain more experience before they become a professor. There's not a direct line between getting your PhD and becoming a professor. So I've been studying in the lab of Dr. John Bell for the past little while, and I hope to be starting my own lab soon. So I'm going to talk to you today about some of the work that goes on in Dr. Bell's lab, as well as some of the research that I've been doing while I've been there. And I kind of modified the title to make it a little catchier, and I'm talking about what I call biological warfare against cancer. And I don't think I have to convince anyone that really there needs to be a concerted effort into cancer research. Most people in this audience has been touched in some form by cancer. And we typically think of cancer as a single disease, but actually it's a pretty diverse group of diseases that are generally characterized by uncontrolled growth of cells within the body. And these are the latest statistics from the Canadian Cancer Society, and they approximate about 171,000 new cases of cancer in Canada this year, and about 75,000 deaths. So this is a pretty monumental issue in Canada, and we're looking for new treatments. If you look at the classical treatments that exist today to treat cancer, such as radiation, chemotherapy, and surgery, they're often really non-specific, and they often result in severe side effects. As you see in the picture, people lose all their hair, people get extremely sick, and it's because of the non-specific nature of these treatments. And so what we're actually looking to develop are what we call specific targeted therapies, therapies that target only the tumor and don't have this kind of carpet bomb effect on the whole host. And just going back to the very basics of what cancer is that really everything that happens in a human is controlled by their genes, right from your very inception, and then in a couple of hours you look like this. Within a couple of weeks, genes determine that you're going to look like this, and then within decades, and keep in mind this slide was made by my boss, not me, you look like this. So I mean, and it's genes really that have a major impact in this development process. And the normal everyday occurrence in most organs in your body, this is how normal cell growth and death occurs. Your cell divides, and when it divides, something else has to die to take its place. So you get a controlled cell death and a controlled cell growth. In the aspect of a tumor, though, this gets kind of flipped on its head. When you get exposed to things like cigarette smoke, excessive amounts of UV, pollutants, and in a lot of cases, just bad luck, what you get is there's a mutation of the genes within cells of your body. The cell divides, but it keeps dividing. The things that control the cell death are actually turned off, and so what you get is uncontrolled tumor growth. And those cells then become immortalized, and then they develop into what's called a tumor. So in normal cells, and you get mutations in a lot of these genes, particularly ones that have to do with cell death, you turn a normal cell into a tumor cell. And it's interesting in our field is that a lot of the same genes that control this cell growth and death also control the ability of cells to fight off viruses. So what are viruses? I don't think this is going to be a huge... Because of the current situation in the news, I think people have heard a lot about viruses in the last couple of weeks, but I'll just basically go over what viruses are generally. Your body's made up of cells, and in comparison, viruses are very, very small. They have no capacity to live outside of the body, and they need the cells of the host to do anything. They get into the cell, they multiply, and eventually they have the ability to spread from the original infected host cell to the next host cell. So really, viruses are parasites of the cell that can only reproduce inside cells. And we talk a lot about viruses, but I think the first point I want to put up to is that viruses are incredibly different. Whereas humans and apes have about a 95% similarity, we're very close genetically. Most viruses have very little or no similarity to each other. These are just 3 different types of viruses that I'll talk about in this talk.
I'm on my way.
And if you look at their sequence identity, they have less than 1%. These viruses are incredibly different, and they have a lot of unique type qualities. So again, I'm going to emphasize that there are a lot of different types of viruses. There are some which get a lot of press that are quite nasty, and they include viruses like Ebola, H1N1 maybe, potentially. And then there are a lot of viruses that are actually pretty benign, and those are the ones that we're going to talk about, and they include viruses like VSV. But before I talk about actually what we do with the viruses, there's an important thing that I have to bring up, and that's how we actually see viruses. As I mentioned, viruses are really small, and most, if not all, viruses cannot be visualized by a light microscope. We'd have to use electron microscopes to actually see them, which is not very feasible for day-to-day lab work. So what we, in the 1960s, we actually were able to take a fluorescent protein from a jellyfish, and the fluorescent protein was known as green fluorescent protein. We were able to take this protein and manipulate it so that we can put it into the DNA of other organisms, including viruses. In the time since we found GFP, we have fluorescent proteins of almost every color. And if you see the picture there of the plate, that's actually drawn with bacteria that express all these different colors. So that gives you an idea of the range of different fluorescent proteins there are. And we can manipulate these proteins so that we can even put GFP into the genome of a mouse. And if you look out on the bottom, this is a nude mouse, and a nude mouse means that the mouse has no immune system, and it also, they don't have any hair. We put in GFP so that if you look at the mouse under UV light, the whole mouse is green, which is really cool actually. But in a lot of the pictures in this presentation, you'll see green cells or green virus. This is how we made it, and it's a very important tool in exploring both cancer cells and viruses, because it allows us to visualize them in real time. So for the last decade or so, Dr. Bell's lab has been focusing on one virus known as VSV, or vesicular stomatitis virus. And it's actually a virus that was originally discovered in crickets. And for the longest time it wasn't characterized, but in a lot of fields where they graze cattle, there are crickets, and the cattle would actually step on the crickets, and they would get the viruses in their hooves, or they'd be eating grass, and they'd get the crickets, eat the crickets, and they would get these sores on their mouth. So it was actually mistaken for a long time as hoof and mouth disease. They eventually discovered, after a lot of panic, because hoof and mouth disease in cattle is actually pretty nasty, that it wasn't that. It was actually this little virus VSV, and it's pretty benign even in the cow. So John's lab has been studying this virus as a potential treatment for cancer because it really has no ability to infect humans, causes a pretty benign disease in cattle, and doesn't even really bother the cricket.